Hi everyone, glad to be here. My name is Dayo, Shittu, and I'm a bioinformatics scientist at the National Human Genome Research Institute at the Center for Precision Health. And today I'll be presenting on the evaluation of the clinical significance of TP53 polyadenylation signal disrupting variants. To start, the TP53 gene is located in chromosome 17 and encodes the tumor suppressor protein P53, known to act as the guardian of the genome. This gene is pivotal in maintaining genomic stability, DNA repair, apoptosis, and cycle regulation. TP53 is activated by both endogenous and exogenous signals, and it is induced as a response to DNA damage when expressed at normal levels. Furthermore, the TP53 gene recruits and regulates several processes that regulate oncogenesis, such as angiogenesis, cell cycle arrest, apoptosis, and oxidative stress. Germline TP53 are known to be associated with Leo-Ferroni syndrome and various cancer types, from colorectal cancer to head and neck cancer. Although not specifically linked to Leo-Ferroni syndrome, the TP53 RS7837822G allele exemplifies the type of genetic changes that can influence cancer susceptibility. This cancer variant is located in the polyadenylation signal site, and it is a rare variant found via WGS in 2011. This variant alters the polyadenylation process, potentially leading to the production of less, production of less stable TP53 mRNA signal, therefore impacting the overall function and stability of the P53 protein and affecting its tumor suppressor activities. This figure shows the age distribution of the two biobanks we use for our analysis, and the All of Us research program and the UK Biobank cohort with the TP53 variant. This shows that the UK Biobank populations are older, while the All of Us carries younger population. Previous research has strongly associated the TP53 variants we're looking at with prostate, glioma, colorectal, and skin cancer, but not breast cancer. Here are two such studies looking at the association of the TP53 variants with cancer susceptibility. Genome-wide association studies was, for the first study, genome-wide association was performed on 16 million SNPs, identified through WGS of 457 Icelander, while the second paper was a meta-analysis on the association of these variants and cancer risk. And both reports an increased odds ratio and potential risk factor for overall cancer. The NIH has helped create the All of Us research program with the goal of establishing a diverse health database of over 1 million participants. The All of Us research program has currently reached over 3/4 of its enrollment and enrollment goal, including participants from across the United States. Most importantly, over 80% of All of Us participants are historically underrepresented in biomedical research, as measured by factors like race, ethnicity, age, income, disability, education, geography, and gender. In particular, All of Us has almost 40,000 genetically inferred African ancestry participants with linked EHR and genotyping data, much more than accessed in previous research. The UK Biobank, on the other hand, enrolled 500,000 participants from the UK between 2006 and 2010, and these participants are predominantly European populations in comparison to the All of Us database. And for this study, this project's objective was to use All of Us research program and the UK Biobank data to identify phenotypes associated with the TP53 of interest and its association with the risk of different cancer types. To do this, we extracted variants and EHR data for our cohort of interest from data sets, performed phenome-wide association study, which is FEWAS, survivor analysis, logistic regression to check the association with specific cancer types, and imputation of variants from the TCGA database. For the first analysis, we performed a FEWAS, and this is similar to genome-wide association study, but instead of starting with disease phenotypes and relating them to the genetic variants, FEWAS starts with the genetic variation and relates it to the phenotype patterns in electronic health record, typically through codes like the SNOMED and the ICDs. Similar to GWAS, FEWAS results are best visualized as a Manhattan plot. So that's what is shown here. Instead of the genetic location along the x-axis, we have the phenotype groupings, and on the y-axis, we have the negative log p values for each phenotype by genotype logistic regression. For our analysis, we found that the TP53 variant of interest with the all of us cohort was significantly associated with benign neoplasm of the uterus and uterine leiomyoma, which demonstrates that a reasonable number of participants with this variant are getting diagnosed. We also performed FEWAS within different groups, and we found that just like the transancestry FEWAS, benign neoplasm of the uterus and uterine leiomyoma peaked in the European and admixed American population. This also shows that the variant presence was more significantly associated with higher risk of certain neoplasms and tissue overgrowth in multiple ancestry groups. Similar to the all of us FEWAS, this result also shows the significant association between benign neoplasm of uterus and uterine leiomyoma. And most of the signals was shown in the neoplasm, citing its association with cancers, especially brain and the skin cancer. And this was done in the UK Biobank. To test for specific cancer types association with the variants, we also looked at the risk of cancer diagnosis. We found that similar to previous studies, TP53 variant in the polyadenylation signal site is highly associated with prostate, skin, breast, and the overall cancer. While surprisingly in the all of us population, survivor analysis only shows significance with overall cancer and skin melanoma, but not prostate, skin, and brain like in the UK Biobank. This is probably because the all of us cohort majorly comprises of younger population and lower rate of cancer occurrences. We also calculated hazard ratio, and the variance is associated with higher risk of prostate, brain, skin, melanoma, neoplasm, and overall cancer diagnosis. One possible reason for this discrepancy is the age distribution difference between these two cohorts. The UK Biobank cohort predominantly consists of older individuals and higher incidence of cancer. We also did analysis using the TCGA data, and TCGA data primarily is a cancer genomic program that produced over 2.5 petabytes of data, described 33 different tumors type, including 10 rare cases based on paired tumor and normal tissue cells collected from 11 patients. And with this data, we imputed our variant of interest. And from that, we did an enrichment analysis for the different cancer types, and it was particularly enriched in adult gliomyoma and thyromyoma, and also in pancreatic, cholangial, rectal, and some non-clear cell kidney cancers. And this shows the survivor analysis with and without the variant of interest, having low probability of survival compared to the IDH that the IDH white type has low probability of survival compared to the IDH mutant, and this variant does not change the prognosis of adult glioma compared to IDH status. Thus shows no significant role in the classification of glioma. And the TP53 classification status is important in prognostication of uterine cancer. However, we have too few people with this particular variant who have uterine cancer to evaluate the impact of the variant in this type of cancer. And same goes for this one, too few carriers to evaluate analysis. And in conclusion, our preliminary analysis demonstrated that the TP53 G allele was consistently associated with higher risk of neoplasms and tissue overgrowth in multiple ancestry groups, and shorter post-enrollment follow-up in the All of Us. The UK Biobank has a higher concentration of participants in the range of 50 to 70 years, with notable peaks around 55 to 65 years, and the All of Us has a broader and relatively more evenly distributed age range. And the UK Biobank, with its older population, has higher likelihood of detecting these associations due to the higher incidence of cancer in the other population. And overall, TP53 variants of interest role in cancer classification and prognostication may be limited. Therefore, further validations might be needed if necessary. Thank you, and I appreciate the All of Us research group and the UK Biobank for making this research possible. Thank you.